I'm super excited to welcome Omar and Jonathan. Omar, I've known, this isn't in the intro, but I'm freestyling. I'm I've known Omar since he was an undergrad, actually, working with Jeff von Molson in my lab. And then he went on to be an MD PhD student. He did his doctoral work at Fangzheng at the Broad, where his research centered on novel CRISPR enzymes for applications in genome editing, therapeutics, and diagnostics. He's received many awards, including the Forbes 30 Under 30 in science and Healthcare and Tech Review's 35 Innovators Under 35. I'm just gonna keep going. Actually, doing 40 Under 40. Yeah. Jonathan received his PhD in systems biology from Harvard, during which he conducted research with Aviv Regev and Feng Zhang at McGovern and Broad, where he focused on the development of molecular technologies for treating and sensing disease states. He's also received many awards, including Forbes 30 Under 30 and Business Insider 30 and Under. Today, they'll be telling you about new molecular technologies for genome editing and cell control. And for those of you who are interested in just creative faculty paths, I think it's also worth talking to them about how they run a joint lab, which is just like something that we're seeing more and more of, but it's still not that common. And I think it has lots of great opportunity to building something together with somebody that you kind of share a brain with. So I'm sure they'd be happy to talk about that. Welcome.
Thanks. So unfortunately, we can't give a exactly alternating word for word talk. So I took the short straw, so I'll be starting today. So I'll do the first half and then I'll do the second half. But thanks for the invitation. It's really great to talk about our work and kind of how technologies, molecular technologies, and technologies can be useful for a lot of different things. So to kick it off, you don't really need to justify genome editing these days, but it is nice to think about how we do have a huge amount of genome sequencing now. They just recently finished the telomere sequence of the human genome, although a little fun fact, that was from a sample that doesn't have a Y chromosome, so it's still in the lamb. But we do have an immense amount of information, and that information demands tools to be able to speak in that language. So you're probably all familiar with things like CRISPR that allow us to be able to search through those 3 billion letters for a sequence which is 20, and then be able to manipulate that, whether that's for detection or editing. Now, of course, this sets the stage for thinking about how other technologies demand new approaches. So now we're actually sequencing many different cells. So the advent of single cell sequencing has really allowed us to understand disease at a new resolution, not at the base resolution, but the cell resolution. And that has been incredibly useful for having a lens to view many diseases and developmental processes, many other things. But it really also demands something when we look at this just projection of the different cells. We don't necessarily know how to manipulate that, how we can do what a knockout would be even. So this begs the question, what is not the genome editing toolbox, but the cellular engineering toolbox? And we have taken a little bit of a crack about thinking about what that is. And it's not necessarily as clean as just being able to target this sequence. So we think about with that having multiple technology. Of course, DNA and RNA are both inside of your cells. Hopefully that's news to you. But so DNA and RNA editing are something that we think about as a component of this toolbox. We also like to think about how we can sense cellular states and react to them, how we can take one cell state to another that we use programming, and then of course doing this in any sort of context, either in a difficult model in vitro or in vivo, we need to actually be able to get any tools we develop into the cells. That would be delivered. Now, unfortunately, we don't have time to touch all of these today, but a lot of us have to think about all of them. We'll be giving a couple of different vignettes on how we can use both RNA targeting, I'll be talking about that, as well as DNA targeting and cell sensing. That's what Omar will touch on today. So how we can be inspired to develop these new tool sets. So kind of as the first vignette, how do we discover and engineer new approaches for targeting and manipulating RNA inside of the cell? And so this is going to be a talk a little bit about CRISPR. So it's helpful to kind of discuss what CRISPR is. And for many people, CRISPR is synonymous with Cas9 or genome editing. But what CRISPR really is, of course, is a whole slew of different adaptive immune systems inside of bacteria. And it's very diverse. So if we just zoom in, little smoother on my computer, but that is where Cas9 lies. So you can see that it takes part of a much larger diversity. And this is nice because, one, it's of course a very interesting system to be able to study, but it also presents us with a fairly large menu of different tools that we can develop. So let's reframe this in a little bit more of a digestible framework. So when we think about how we can discover and engineer new CRISPR tools, it's nice to classify them into two classes. So we have hopefully this you can be seen online, but we have the class 1 tools or systems. We have the class 2 systems. And you might be able to tell this, but what distinguishes these is that in the class 2 systems, the many different functions of CRISPR, which is involving cutting nucleic acids, binding that programming molecule, are all consolidated into a single protein. Where in class 1 systems, those activities are actually distributed among many different proteins that participate in a multi-protein ribonucleic protein complex. So you can imagine that these are probably not as useful as tools because you have to constitute many different proteins inside of the cell. While here, Cas9, and CBF1, which is also known as Cas12a, those are the more popularly known tools because they're much more easy to use. So we can still mine these and discover among these. And we can also use commonalities like the Cas1 enzyme, as well as, it's not on here, but the CRISPR elements themselves to discover new tools. So we've been doing this for quite a while. So now over 5 years ago, we actually discovered a new CRISPR enzyme called Cas13a. And it functions very similar to Cas9. You can program it to target nucleic acid, but it targets RNA instead of DNA. So this is actually potentially quite useful for obviously manipulating RNAs inside the cell. We can knock them down, work with factors to them, which allow us to study their function. And of course, just to elaborate on that, there's a lot of different RNAs in the cell. There's obviously mRNAs, non-coding RNAs, tRNAs, rRNAs. And being able to target each of these individually in a program sense allows us to actually be able to study the function. However, there are some caveats. So Cas13a has this activity, it's called the collateral activity. And we won't go into it too much today, but it basically means that when it binds to an RNA, it becomes activated and cuts other things in the cell. And we call that collateral because you're not just targeting what you want to target, you can actually hit other things. And in certain cell types, especially this is the first cell type found in UAD7 glioblastoma cells, when you target an RNA, here you get reduced cell growth, so you can see that there's actually fewer cells, and you actually get degradation of the endogenous RNA. This is a gel showing the ribosomal RNAs inside of these cells is actually becoming degraded upon targeting of this other RNA transcript with EGFR B3. So this isn't great. If you want to be able to study the knockdown of a RNA, you don't want to be able to have an adverse effect on cell health or other non-target RNAs. So when I started our lab, we really were thinking, okay, well, we are at a point where we have RNA targeting tools that come from natural CRISPR systems, and those are kind of meeting the mark. But can we actually find new ones that allow us for better transcriptome perturbation by going back to the drawing board? Now, this is where this CRISPR diversity comes into play, because obviously the class 2s are great. They allow us to put one single protein in, but the class 1s are cool too. The type 3 systems, for example, they are known to target RNA. So if we could turn them into an RNA targeting tool, that would be perfect. However, they have these multiple different proteins. Not perfect. But as is in the case of evolution, sometimes these classifications, that class 1 and the type 3s have multiple different proteins, may not be universal. So we were at a conference in 2019, I think actually the last conference we went to before the pandemic. And we saw a talk from Kira Makarova, one of our collaborators. And they described the diversity of these type 3 families that are known to target, you can see RNA targeting, RNA targeting, RNA targeting. And they pointed out this very interesting enzyme here, which is a type 3 family member. It's the type 3Es. But interestingly enough, it wasn't multiple proteins. It looked like all these different proteins kind of been fused together into a single polypeptide, which would be really great if we could then put this into cells and use it as a tool. Now, of course, this was all computational, and we had no idea whether this actually targeted RNA or would work in mammalian cells. So we began a bit of a combined biochemical and microbiological and cell biology search to understand what this protein was. So we actually did a computational expansion and found all these different family members, which we call the Cas711s. It's a little bit tongue in cheek, but it's because it's multiple different Cas7 domains, which are kind of the scissors in this context, as well as a Cas11 domain, all fused together into one protein. And they do seem to occur like that in many different contexts. They're also similarly related to a more old intermediate, which is multiple things fused together, 3 Cas7s fused together. But we actually found as an aside that didn't cut RNA. But we found many of these different family members and we asked, could these actually be programmed to cut RNA? So we actually purified the protein, which is a fantastic way to get it down to its most basic targeting aspects. And we showed that when you program it with a guide RNA, CRISPR RNA against this target, you could get this cleavage, which is really fantastic. It was incredibly precise, so we needed a targeting RNA, the protein, also cations. And then we decided to actually tie up CRISPR RNAs across this target and see what it looked like on the gel. And the really amazing thing was that we actually saw that these bands, and now there are 2 bands because it basically comes in 2 places with the product, these bands shifted in their position, which is fantastic. It's what you would expect. But if you do this with something like Cas13, that collateral enzyme, you don't see this because it's cutting everything. So this is a really amazing validation that we think that this is working in a way we can program it very precisely to cut in 2 places on this RNA and be able to have no potential off-target effects. This is great. Now we wanted to say, can it be used in mammalian cells? And this is always kind of a nail biter because when you're characterizing a protein, you have no prior. You can only try your best and find some orthologs. But we decided to just go for it. And we used a assay that we like to turn to a lot, where we use 2 luciferases, a Gaucher luciferase and a cryptogenoluciferase, and we can target one of them and measure out the ratio as a really easy way to screen an RNA targeting enzyme. And so what we would want to do is target this GLP and be able to see we're getting reduction. And we actually were really surprised, partially, but relieved, I would say, to see that we could actually get a great reduction. So we compared Cas711 to RNAi as a kind of old school RNA targeting technology, as well as several of the Cas13 family members, which now there are about 5 or 6. And we saw that we could get levels comparable or better than the other competing technology with Cas711. So this is really fantastic because it works in mammalian cells. It seems to be quite effective. And obviously you don't want to just screen on reporter construct. So we actually targeted a panel of different targets, KRAS, PWD, as well as the non-coding RNA intolerant. And we saw that to different degrees, mallat being a little bit less effective, we could get fairly good levels of knockdown with Cas711, especially when compared in guide control fashion to different Cas13s. So this was great. We can see that it works. Now we wanted to kind of increase the difficulty level a bit and look at the collateral activity. So how can we actually detect collateral? Well, there's a couple of different ways that we can look at this cutting of other things in the cell. We like to use this in vitro assay. So here we can actually detect in a test tube this cutting action by doping in a fluorometric reporter, and that gets cut. And here, when we compare Cas11 to Cas13, we can see when we target a RNA with Cas13, it shoots up, allowing, this is indicative that it's cutting that other non-targeted fluorometric reporter. Cas11 stays completely flat. So it has no actual cutting of these other things inside of this test tube. Then we want to go up to bacteria. So this collateral activity actually inhibits cell growth, and it's actually a defense mechanism. So if we do this in bacteria, we should see an inhibition of cell growth upon collateral. And that's what we saw. So we actually targeted RFP with either Cas11 or Cas13. And you can see in the case that we target RFP with Cas13, it actually substantially inhibits the cell growth of these bacteria, but only in the presence of target. And with Cas711, you don't get any growth inhibition either with or without cell target. So this is great. It has no function of collateral in either test tubes or bacteria, so we can be totally confident that it will have no effect on cells. Well, let's see. That's always nice to hope. But First, we wanted to look at the U87 cells. So these are the glioblastoma cells that we introduced earlier. And we concentrated targeting with Cas711. This is just knockdown and Gaussian luciferase. And you can see that it works, once again, better than 13A. As a little bit of an aside, RNAi does not work in U87 cells. They lack, I believe, a risk complex. But this is awesome. It knocks things down. What's the toxicity? So we plotted, once again, the expression, so this change in expression versus a change in cell viability. And you can see with Cas711, one, just without targeting, it's most healthy. But two, we have no change in cell viability. Versus many of the Cas13s, for example, PSP Cas13b, where you get non-targeting, which is already toxic, down to an increase in cell death from Cas13. So you can actually see that compared to the other Cas13 tools, Cas11 is a much less toxic approach. So we do this as one of the many components of this spectrum of RNA targeting tools. So of course, there are many different applications we can use. We can use them to modulate translation, modulate splicing, alter localization, and even modulate epitranscriptomic modifications. We're especially interested in how we can use the precise cleavage of Cas711 for actually being able to very precisely modulate and replace information using splicing. So we're actually doing many follow-ups. So one thing is we've actually determined structures of Cas711, and we don't have time to chat about that today, but that story should be coming out pretty soon. And it's quite nice. We can actually use it to decrease the size of the protein. We're continuing to optimize it for in vivo applications, including packaging it in a single vector for in vivo delivery. And then we like to use this precise cleavage, allowing us for, as I mentioned, the ability to not only knock down RNAs, but to change the information via modulation and splicing. And these are all part of what we imagine as this RNA targeting component of this cell control toolbox. So we do it as a really fertile area for allowing us to modulate the transcriptome, and we hope that it'll be adopted as well. So that's a lot of the things we can do on RNA, but of course, there's also DNA. So with that, I'll pass it over to Omar so he can talk about the work we've been doing on how we can manipulate not only the transcriptome, but the genome. Cool. Awesome.
Yeah, so I'll sort of close the talk with two other technologies that I'm working on. The first being a way to do programmable insertion of large pieces of DNA. And then I'll end on some of the cell control stuff that Jonathan mentioned in the beginning, to how they actually sense and respond to different cell states. So just to dive in, as I'm sure many of you are aware, the sort of genome editing toolbox is getting quite mature. And Cas9 was, of course, amazing for being a RNA-guided programmable nuclease, and it was then repurposed as a way to kind of just GPS locate any piece of the genome to recruit different effectors, like transcription factor activators for modulating gene activation, deaminases for doing editing, and even most recently like reverse transcriptases to do more versatile edits via prime editing from David Liu's lab, and of course the sort of larger transposase and recombinase systems, which people are still trying to get to work in mammalian cells. And so, when you think about all the functions you would want with a mature genome editing toolbox, we're starting to get pretty close to every possible thing you can imagine, with one notable exception, which is the ability to do large insertions in non-dividing somatic cells. So, early on, people thought, Cas9 would be great because you can cut and then use repair pathways like HDR and non-homologous end joining to do things like any edit. But it turns out in vivo, HDR is actually not that active in a lot of cell types. And so that becomes a challenge. And of course, with the most recent prime editing, you can kind of do insertions up to 30, 50, 100 base pairs. But you certainly can't do full genes. And why, well, before I get into why that's important, I guess I'll describe kind of one of the solutions we've been toying around with for a while, which is the recruitment of integrases to do this. And so the thought is, if there are enzymatic components that can load large pieces of DNA into the genome, can we repurpose those to do that? And so integrases are a common one. People are probably familiar with recombinases like pre-recombinase. Now, the challenge with these is it's hard to separate their catalytic activity from their DNA binding specificity. So it makes it really hard to program them. And what we realized is when prime editing came out from David's lab, we realized that even though it's limited to 50 to 100 base pairs, that's actually the perfect size for the landing sites for a lot of these enzymes, like BXP1 integrase, 5C31, or so on. And so we came up with a system where we could actually fuse an integrase to the prime editing system and use the guide RNAs to actually lay the beacon for these integrases into the genome, and then simultaneously have the integrase recognize that beacon, which is about 50 base pairs, and load a larger sort of insert into that. And so you're left with two scars from the insertion process, which are each about 30 to 50 base pairs, and we called the system HASTE. And so, one of the reasons we were really interested in developing this HASTE system is because actually a lot of mutations are not driven by, a lot of diseases are not driven by a single mutation. So this is the spectrum of the number of mutations in different liver and eye diseases. And it turns out that, often you have dozens, if not hundreds of mutations. Cystic fibrosis is another example where there's 2,000 different mutations that are in patients. And so when you think about developing like a base editing or prime editing drug to actually address all patients, you'd have to make 2,000 cystic fibrosis prime editing drugs if you actually wanted to treat everyone. Now with gene insertion, you would have a single drug. You just drop the gene in and it applies for all the mutations. And so that's kind of the advantage if you can figure out how to do programmable insertion in vivo. It's kind of bringing back to the roots of gene therapy, which is actually like the replacement of the gene, but in a way that's actual, programmable. It's at the normal genomic sites. You get the promoter that normally regulates the expression of these genes, and so it's kind of better than just overexpressing a gene that eventually gets silenced with time. So yeah, so to start out, we had originally actually tested pre-recombinase because we thought naively that would just work. And it turns out recombinases are actually not that good at inserting into the genome. It was very low efficiency, like maybe 1 or 2%. And we realized that recombinases are actually much better at unimolecular reactions where you're recombining, two sites on the same strand where it just inverts it. But it's not good at two separate molecules that you need one to go into the other. And so we then looked to another, so these are tyrosine recombinases. We then looked at serine recombinases or integrases, which are from phage typically. And we tested a panel of those, and we were pleasantly surprised to see much better activity because that's what they've evolved to do, which is insert a phage genome into a bacterial host. And we ended up sort of focusing on BXP1 because it was the highest efficiency with this sort of prime system. And so we then did a whole campaign of engineering to kind of link the integrase to the prime editor, different linkers. We did different mutations that improved the processivity of the RT, and we tested a few different endogenous sites with different guide scaffolds. We found, I think, a certain version F here, or F that was giving generally the highest distribution editing, and we went with that, which we called PASTE-V3. And we found in general it was pretty good at loading various size DNAs up to 13.5 kb on this graph with two different sites and 36 kb in this plot of DNA to the genome. And actually, I think there's a little bit of a decrease here. This is more of a challenge of getting the larger pieces of DNA into the cell versus we think kind of an enzymatic limitation. Because normally these integrases integrate like 50,000 base pair phage genomes into bacteria. We also tested a whole range of different endogenous sites in the human genome and compared it to HIDI, which is basically just a fancy way of using non-homologous end joining to insert a gene into the genome. This is more common. As I'll show, non-homologous end joining has a lot of issues, like it can insert the gene in two different directions. You can actually control how it inserts. It has a lot of indels, but also in general, we found PASTE was more efficient than relying on non-homologous end joining. And as I mentioned, HIDI has a lot of indels because it's a double strand break based method. So with HIDI, you get some integration, but it turns out actually the majority of insertions are indels, as shown in the white bars. With PASTE, because it's a dual nicking based approach like prime, it's actually much higher purity. So most of your edits are insertions and either very minimal indels or manageable indels compared to your integration. Similar to HDR, of course, people know this for HDR, but usually you get a ton of indels with HDR as well, compared to your integration, whereas with PASTE, although it's comparable efficiency, it's lower indels. Of course, HDR won't work for non-dividing cells. One, I mentioned this at the beginning, but one criticism of this approach is there's two scars on either side of the insert, which can limit, if you need to do in-frame gene tagging, for example, or in-frame applications. And generally for therapeutics, that's less of a concern because you're probably inserting the gene in an intron or promoter where it doesn't matter if there are scars, but we just wanted to show if we picked different genes and tried to fuse a GFP to the gene and use the SCAR as a linker, it was possible. So the SCAR can actually be used as a protein linker. And yeah, thanks for lowering the lights, but basically the paste is the green and the red is an IF stain, and the different localizations co-localize lamin data. You can see the rings around the nucleus. And for NALC1, you see kind of the nuclear sort of punctate form. So just to show that you can use it for in-frame if you really need it. So I think that's it for images. Thanks. Yeah, one other application we just want to mention for paste. There's actually a lot more in the preprint that we don't have time to talk about today, but it's cool because the integrases have they have flexible attachment sites, but they're very, the payload is very much connected to the sequence that is complementary to it in the center. So it turns out that there's two bases in the center of the beacon attachment site that if it's complementary to what you're laying in the genome, it connects the payload to that beacon. But for the same integrase, you can change those two center nucleotides and it allows you to basically do multiplexing where you can specify, for this beacon, put this payload there, and for this beacon, put a different payload. And it allows you to do easy multiplexing with the same system, which could be useful for cell engineering applications. We actually characterized a lot of this sort of attachment site flexibility. And it turns out for certain dinucleotides in the center, there's actually really good orthogonality with minimal crosstalk between the beacon and the donor sequences. And that allows us to then, do all sorts of combinations of multiplex gene insertion. So 3 different endogenous sites, 3 different cargos, and it works pretty well. And so the hope is, maybe we could, we're starting to explore this for T cells where you can load different things into the T cell. This potentially could be useful. So one other thing we explored, so I've mentioned the non-dividing aspect now. So afinicoline is a cell cycle inhibitor that inhibits the cell cycle at the G1S phase, and it basically inhibits HDR. So you can see EMX1 based HDR drops by like 95%. But with paste, we actually don't see a drop in efficiency, just to show it's not really reliant on cell cycle dependent repair pathways, much like HDR is. And we show this at 2 different payload sizes, just to show the versatility of the paste system in non-cycling cells. And I will have some sort of somatic non-dividing cell data as well, which we've explored in vivo a little bit. So just to show a quick early look at the data, we developed a viral system to deliver the system. It's quite large, so we have to split the system across 2 adenovirus vectors, and then we use a 3rd vector as the payload. And then we can inject into mice and look, 4 weeks later for editing. And we actually used humanized hepatocyte models from Eucurus to look at this. And this is like just a very early experiment where we haven't done much optimization yet, but we were happy to see at least that there was some editing in the liver, and then the negative controls, of course, were negative. And so now we're trying to optimize for better delivery and better doses, but it can work kind of in sort of non-dividing hepatocytes. So cool. Oh, and lastly, when you look at those, there's actually sort of decent purity of integration with low wild type indel formation, as you would expect from the in vitro data. So, yeah. So in general, I think we're trying to optimize the in vivo delivery. I think we have a lot of mRNA-based data versus viral delivery in the paper in vitro, and now we're trying to take that in vivo to do like mRNA lipid-based delivery because that in general will be better and safer. And we're also trying to apply this to a few different diseases. I think cystic fibrosis is a really interesting one just because there's so many mutations in the patient population that this could be helpful for. And of course, I think we're also, this type of tool can be useful for also just high throughput gene tagging and other sort of basic biology approaches as well. So we'd definitely be excited to chat with anyone about paste if there's interest. Cool. So yeah, as we mentioned in the beginning, a more recent technology that we actually put on bioRxiv in January was to basically respond to cell states in sort of a way that allows you to output different proteins. And so the kind of motivation here is, Cas9 kind of came about from the sequencing revolution, right? We understood mutations can drive disease and other phenotypes in biology, but we didn't really have the right tool set to actually study those mutations and then potentially treat them. And we felt like something was happening similar to the single cell RNA sequencing field, where you generate all this data and you can say, hey, there's 20 different macrophage states involved in whatever, inflammation and cancer and autoimmunity, but we don't really have tools to then say, hey, what if I want to downregulate state 3 and see if that is actually what's causing this phenotype for state 5? And so we wanted a way where you could actually sense a single RNA marker, combinations of RNA markers, and then do something based on those in sort of a live cell and in a non-destructive way. And the basic idea was that if you had a sensor that could input some sort of combination of RNA markers, you could then express any protein. And so it was funny because we were exploring a lot of different ways to do this. And, I think Jim Collins had a paper he published in the fall and trying to do this with like 12-fold sensors in mammalian cells, which is like a different way of thinking about that problem. But we had spent a lot of time in the RNA editing space for therapeutics. And just briefly, the concept there is you want to recruit ADARs, which are like RNA deaminases, to transcripts, and you can actually use them to correct mutations. So like ADARs will do A to G changes, and it's a really powerful way for RNA-based editing therapeutics. And what we realized is we can maybe flip that paradigm on its head. So instead of actually trying to edit something in a transcript, use the transcript to force editing of the thing that's binding. And so the idea here was if you had a sensor, and the green thing is like your protein you want to express, like a GFP or a luciferase or a caspase killer cell. What you do is you block translation of it by adding a stop codon upstream. And you put it in a region, aka a guide, that's complementary to an endogenous transcript. And so when that transcript binds, if you designed your guide in a way such that the stop codon is across from a CCA, so that the A inside the stop codon is actually forming a bubble across from a cytosine, which is the preferred deamination site for ADARs. The ADAR can come in and actually edit that stop codon to a UIG, and that causes read-through because that's read as a UGG tryptophan. And so then basically you can then get translation of your output protein. So it's a very programmable way to connect the transcript input to the expression of a protein. And you can either supply the ADAR exogenously if you really want high editing, or you can actually design these guides in a way such that you can recruit endogenous ADAR as well. Because a lot of cells, actually almost all human cells, express ADAR1, and ADAR2 is highly expressed in the brain. But ADAR1 is actually what we spend a decent amount of time trying to optimize for. So there's of course a lot more data in the paper, but I'll just cover a few highlights. So we designed an initial very basic sensor against EGFP, and then the output was luciferase. And so in the presence of target and in the presence of sort of the overexpressed ADAR2, you actually get like 50-fold activation of luciferase. And actually, if you don't supply ADAR, you can still get like 5-fold activation, which is the endogenous ADAR kind of helping to activate a bit. And we actually then switch the sensor to an EGFP output. So you could sort of do flow or sort of single cell analysis. And similarly, in the plus target condition, plus ADAR condition, you can get really good sort of output of the sensor. And then this is harder to see here, but there's very sort of lower levels of fluorescence in this square due to endogenous ADAR. If you look at the editing matches. So when you have target, you get sort of UAG to UIG editing from the ADAR. And we did a lot of sort of guide optimizations to try to boost, the activation. So there's two things you're fighting against. One is like guide designs that really maximize your RNA editing levels, but and one thing that helps with that is longer guides. So like 100 base per duplex, 200 base per duplex. Problem is if you start to get into longer guides, you actually can get read through of your stop codon. The ribosome will just express your sensor. So you have to then do things like add MS2 hairpins to kind of block the ribosome a bit. So there's a lot of like tricks and things we can do, like out of frame stop codons to also catch the ribosome. But basically using those tricks, we can get the activation up to 150-fold, and now in our lab we're actually doing 1,000 to 2,000-fold activation, and even more tricks. So I think there's a really rich engineering space here that's less interesting if you just want to use the tool. So I'll skip over a lot of it, but some of the designs we had were actually quite quantitative, so you can map the radar's sensor output to sort of input qPCR based gene expression. So there's some quantitative nature to it. We tested endogenous sensing, so that earlier data was based on reporters. Endogenous is actually really hard because you have to basically modulate the transcript, so it's less easy than a doxycycline-induced reporter. But to do that, we used siRNA basically to sort of modulate an endogenous transcript and see if radars responded. And so these are two different transcripts we targeted. You can see the PCR response to the siRNA, and then the radars also has a knockdown response as well. So that shows that we can do some endogenous sensing. We also tried a version of the output where it wasn't for tracking, but actually to do something. So we expressed the caspase 9 based off of the sensor, and we just show we can get kind of a 70% increase in cell death using this system. And of course, the constitutive caspase doesn't really have an increase because it's constitutive, but the sensor kind of has a nice increase for no target. And one other thing that was kind of neat that we're optimizing is the ability to do AND gates, because it's such a simple modular system. You can just string the guides together. So if you have two targets, you'll have to have both stop codons edited based on the presence of both targets. And so when you have both targets, you get a higher fold activation. We've actually, with newer optimizations, been able to get this like 50-fold with no background from individual targets. So that was kind of a nice development. And I think there's probably some limit. I don't think we'll be able to do this for up to 10 transcripts, but we're exploring now 3 or 4 transcripts and seeing how far can we push the limit for these AND gates. And lastly, just to show a little bit of preliminary in vivo data, one of the cool concepts for radars is, controlling mRNAs, right? So in the virus world, you have cell-specific promoters if you want to target, only hepatocytes or specific lung cell. But with mRNA, you have no way to control what cells they express it. Once they get the cell, they express. And so radars is kind of cool because you can just tag it, the upstream part of your mRNA with this guide, and then you have a way to now kind of say, hey, only express in this cell type. And so we wanted to show this. We actually made synthetic mRNAs. We put the radar's guide upstream of the ACA loop, which is like a deep red luciferase for in vivo imaging. And then we just got some mice that expressed human serpin A1. This is the PIZ mouse, and wild type mice that don't have human serpin, so we can design the sensor against human serpin. And then rely on endogenous ADAR for activation of the sensor, and then we can just basically image next day to see if the sensor is kind of outputting the luciferase. With this kind of, again, it's very preliminary, but so if you have a ACA-loop mRNA that's constitutive, you see expression of it in both mice. If you have one that's a non-targeting sensor, there's minimal expression. And then with 2 different sort of targeting guides, we saw some cell selective or strain selective expression of the sensor between those. And it's like 2 to 3-fold activation. And now we're doing more optimizations, trying to get better mRNA quality and use actual LNPs versus this like commercial reagent to deliver these mRNAs and try to see how far we can push the fold activation. But I think, yeah, it's a sort of work in progress to see how we can use basically this control process for cell selective mRNA. So in general, we're continuing to make improvements. We're trying to do, screens to identify like how to actually design these radars guides. I think there's really interesting applications in both the cancer and autoimmune space that are worth exploring. For example, in cancer, can we use this to only express cytokines in the cancer cells to heat up the tumor microenvironment in a way that not only hits the primary tumor, but like metastases? And that's, I think, one area that's where we're currently trying to look at. And of course, it could also be used for in vivo, biosensing applications. Like use this to sense, 5 different markers involved in immune cell response to a tumor, just even tumor-specific transcripts. One thing I also didn't mention is you can also use this to detect specific mutations in the transcript as well. So like the ADAR is really specific to mismatches and kind of the deamination bubble. So you can take advantage of that for example, cancer mutations. So yeah, I think it's really early days for the platform, but a lot of maybe interesting directions. Yeah, with that, I think in general, the way our lab thinks is, are there enzymes, other things in nature we can take advantage of to basically build tools, from integrases, from phages to sort of these ADARs and whatnot. So we're always recruiting. Anyone's interested, and thank you to many members of the lab that drove these projects. Kai was really behind the radars work, and Matt was behind the paste work, and Austin was really driving the Cas 711 work. She's no longer in the lab now, but and our many collaborators. So yeah, thanks.